Start the recording. Okay, so welcome everyone to the second session of our BioClock Academy lecture series. My name is Wana Georgiana Rus Oswald, and I'm a postdoctoral researcher in the BioClock Consortium. And together with Hannah Brooks, which is a PhD student in the same consortium, we will host the session today. The BioClock Consortium is a Dutch-funded organization which aim is to study the biological clock in 3 different levels: health, society, and environment. And the BioClock Academy is a lecture series which is organized within this consortium, and it is addressed to anyone which is interested in the field, especially early career researchers. And the aim is to introduce you to the basic concepts and to keep you with the proper knowledge for your research. And we invite every third Wednesday of the month, we invite different experts in the field. About the time frame today, we will have 40 minutes of talk, followed by 15 minutes of discussion, so roughly 1 hour. And during the talk, you can write your questions in the chat so that we can discuss them at the end of the talk. We will keep track of the questions. And with this, I want to give the floor to Hannah, which will introduce our speaker today.
So hello, everyone. Also a warm welcome from me. It's nice to see that you're all here for the topic of today, which is about the importance of biological clocks in species ecology and for their interactions. The talk will be given by Kamiel Spoelstra, Dr. Kamiel Spoelstra, who I would, of course, like to welcome. So Kamiel is a researcher at the Netherlands Institute for Ecology. He did his own PhD on chronobiology under supervision of Serge Daan at the University of Groningen. After that, he also did several postdoc positions, also on the topic of chronobiology and also on light pollution. And he continues this work today with his own Spoelstra lab, where a lot of the research is focused on artificial light at night and how this impacts the temporal and spatial activity patterns of species. So both on population level, but also on species interactions. And over the years, bats have gained a central position in his research, but he keeps involved in a lot of different projects. So it's just incredibly interesting to talk with him about these topics. So I'm sure we will have a very interesting lecture today. So yeah, with this, I would like to give the virtual floor to you, Kamiel.
Thank you very much, Hannah and Georgiana. And it's a pleasure for me to give a talk in the series of the BioClock Academy. I would like to share my screen, and I need permission from the host, I see.
You should be able now to share your screen.
Yes, that works. Okay, so if everything is all right, you are now able to see my presentation. Is that correct?
Yes.
Can you all hear me very well?
Yes.
Okay, very nice. Okay, then I'll start. Just put my clock as well. So I'm going to talk about clocks, but I need a small clock myself when giving presentations. That's interesting by itself. But clocks in ecology, yeah, and I think that's truly a phenomenon that we should care about. And I hope at the end of my talk you will fully agree with me. To tell you a little bit more about myself, if everything goes right, then you should see a different slide now. So this is me standing somewhere outside in the field, listening for bats. Hannah already told it. Already as a teenager, I was fascinated by nocturnal species, and as such went out at night really a lot. And yeah, indeed, I worked a lot with bats, also with mice and other mammals. But I always had the desire when I was in high school already to identify bats, and I quickly found out that this is actually quite difficult. So for nights and nights in a row, I sat in the basement of this old castle ruins close to my parents' place, trying to photograph bats in full flight in order to identify free flying bats. And I had to find out really a lot of things, which I won't be talking about. But I built whole setups to catch these flying bats, and eventually I succeeded in photographing bats, and I was just totally happy. But I think that's what my fascination for nocturnal species comes from. And by the way, at that time, I also found out that I myself, like most people, I am a diurnal species, and you compromise your own health a little bit when you try to reverse your own rhythm. Okay, so rhythms in ecology, that is really nice topic to talk about. But it's really broad, and as you may know, rhythms in ecology, there's rhythms just virtually everywhere. So I won't cover all of these rhythms today. I will mostly focus on daily rhythms and yearly rhythms, although I'll mention some others as well. And of course, everybody of us is really acquainted to daily rhythms. As I just told, we like to be active in daytime. But if we go at night, we hear owls and we can listen for bats. And virtually all organisms in the planet, the planet's been rotating for over 4.5 billion years. Virtually all organisms follow a day and night rhythm. So these are really hardwired and really pervasive. Yearly rhythms, of course, because the pivoting Earth are also super rooted in many ecological systems. Here I have a small diagram. There's a tree in there, but actually it's about migrating birds. And if you've been looking outside, then you may have noticed that about a week ago, the last swallows actually left. Hey, that is a bit weird. Left to Africa. All right. But there are many other rhythms, which I briefly will mention. Of course, here, let's see whether you can see my mouse. Let me see, where's my mouse? Can you all see my mouse? I guess so. So here we have, of course, the circadian annual rhythm, as just talked about. But of course, we also have the moon orbiting Earth. There's a lot of species that actually respond to the lunar cycle. Also species that respond to the half lunar cycle. And of course, there's strong tidal rhythms. And of course, as you can see, many marine species are strongly tied to circotidal cycles, which are strongly linked to the moon. And that, of course, is very logical. But there's also lots of other organisms that actually respond to the moon cycle. For example, nocturnal mammals, a lot of insects and bats. If you go out at night with full moon, there is a lot less bats to hear as opposed to when it's very dark. And there's lots of much more extreme rhythm. For example, you have cycads in the US that have a 15 to 17 year cycle in reproduction, depending on the average temperature and the habitat they actually live in. Okay, so before I go any further, I would like to explain you the sort of standard graphs we all use to depict activity rhythms. Because I'm going to be showing a lot of these. So what you can do with animals, you can put them in a running wheel. As running wheel, you can actually keep track of the number of revolutions of these running wheels. You can keep birds in a cage and then put a switch on the perch. These birds actually hop on and keep track of the number of clicks of these switches. Or you can inject an animal with a microchip, for example, and record how often such an animal actually passes a reader. Now, if you would do this and you would plot these events, either clicks or revolutions or readings, on a line that runs from midnight to midnight, and each individual record will be a tiny vertical line here, then you get an activity record. And each following line is a following day. So if you plot these lines beneath each other, then you get a very nice impression of the rhythm of an animal. Here on this side, you see the rhythm of a mouse in a light-dark cycle. So here it's light, this is dark, and here it's light again. And you see that this mouse is actually active in the dark phase of the schedule. If you do not apply a light cycle, then most organisms revert to their endogenous circadian period. So their own internal clock starts ticking and tells these animals when to start and end their activity. And as you can see, this mouse here starts a little bit earlier every cycle it processes. So you see this is not the 24-hour rhythm, but a slightly less than 24-hour rhythm. So I'm explaining you this because I'm going to show you really a lot of these rhythms, and you have to understand these rhythms in order to understand what I explained to you. Now, what people often do is double those rhythms. So just make a copy, just like let the time continue. So in other words, this cycle here is the same as the cycle there, and this cycle here is the same as the cycle there, and the same for this actogram as well. And you can understand that doubling this actogram is actually really useful if you have rhythms that are really short and actually wander from one time frame to the other. Now, to get back to circadian rhythms, to tell you a little bit more about these. So I just showed you the rhythm of a mouse in a light-dark cycle, where the mouse is active in darkness. Here's the rhythm of a human. It looks actually a bit more sloppy than that of a mouse, and that has to do with our own behavior. We have alarm clocks. One morning we get up a bit later than the other. We have clocks in the evening, so one day we get to sleep a little bit earlier than another day. Anyway, we have these rhythms, and in a light cycle, they actually follow. If you average them over a long time in a normal light-dark cycle, they follow 24 hours. Here you have, so these are circadian rhythms. Here there is also circadian rhythms, even though it says non-circadian, and you can see them actually here. They're much more vague here. So here is it's not a mouse. This mouse has a long tail and this has a short tail, which means this is a vole. It's a whole different animal. This vole eats much less nutritious food and needs to forage much more often than this mouse. So actually it has many more activity bouts. So this mouse actually prefers to be active at night. So it's a bit more active in this part here. And it's less active here in the light, but it's active everywhere. And you can clearly see a circadian component here because it links to darkness here. Same for this reindeer. The reindeer is a grazing animal. It's maybe not all too afraid of predators, can graze all day, but still there's a circadian component here. But the other components here, like the intermittent feeding activity here, and as well for the reindeer here, we call non-circadian, most of the times called an ultradian, so faster than a circadian, faster than once within a day. Now, if you, this is a light cycle, so if you deprive organisms from a light cycle, if you lock them up without any light information, without any time information, they revert to their endogenous circadian rhythm, which I already explained to you in the previous slide with the mice here. We humans do that too. We actually lengthen, on average, we lengthen our rhythm a little bit. But look at this mouse here. This mouse has this ultradian rhythm here. It gets a super ultradian rhythm here because it's in darkness. It doesn't need to bother about light or darkness, and has greatly shows this rhythm here. And here, this reindeer also fails to show any circadian rhythm. It's not relevant anymore. There's no light and dark information. It can forage anytime it likes, and it doesn't need to bother about light and darkness. There's actually, so these rhythms are a bit more extreme. These are organized according to a light-dark schedule. These are less organized, but well, they are organized from a system within the animal. That's important to keep in mind. But there are also other rhythms that are sort of superimposed by the ecological circumstances that animals find themselves in. And I'll show you, and don't try to grasp all of these rhythms. It's just like one big image of many different waders, some in the Arctic, some not in the Arctic, with which I want to say some are in constant light. As you know, in the Arctic summer, there's constant light. There's a sun going a bit higher and a bit lower, but it never sinks. And some are still in an area where the sun actually sets. But anyway, these birds, they have to breed, they have to incubate their eggs, and they share, they take turns on this. So the male and the female take turns. And what happens here is that they have their own rhythm in incubating. So if one of the sexes actually is incubating, the other only will take turn if the other leaves. And in other words, they sort of get an assembly of two different rhythms, which actually compose completely different rhythms, in many cases, completely different rhythms from 24-hour rhythms, which doesn't mean a lot of other rhythms within these animals actually are not tuned to a day-to-day base, but at least these incubation rhythms are extremely various, and they are completely driven by other ecological motives from these birds. And I have to say that's mainly predation, but I'm not going into detail. Now, so you may say, well, okay, if rhythms are superimposed or if rhythms are derived from life cycles, why do we need a clock, right? The clocks are extremely important for stabilizing our rhythms and also for anticipating events. And one of the nicest examples of this is the European ground squirrel. It's a European mammal. They live in southeastern Europe. They're strictly diurnal. They burrow underground, and they only emerge in daytime when the sun is already up for hours and there's lots of ambient light. And if you put these ground squirrels, if you fit them with a light-sensitive collar that knows when they're above ground or when they're below ground, you can actually also make an actogram. And that looks like this. So it's an actogram provided by Roelof Hut, also a prominent member of the BioClock consortium. So here, this is an actogram. Every consecutive line is 24 hours, in which Roelof plotted the night and the day. So like when the color changes from gray to white, that means that dusk and dawn are exactly at those places. And as you can see, the activity recording goes from early spring to early summer, and you can see the day length actually increasing. What you also can see is that these animals actually are very, very accurate in emerging from their burrows. So they really emerge. They don't make mistakes there, right? They are only above ground hours after the sun is above the horizon, and they submerge in they retreat in their burrows hours before the sun actually sets. And their clock must play an important role there because the light information is very subtle there. So so these animals, they can very, very well use the clock to anticipate the right moment to emerge above ground and to retreat. Interesting, so it's funny, it's a bit of an anecdote. So when I just was a PhD, there was a solar eclipse in Austria, and we went there and we did the same. We recorded these ground squirrels that were above ground, close to Vienna. And we did it for 3 consecutive days. This is actually an actogram. It's only 3 lines in a row, right? So here you have manual activity counts, and here the black lines here are radio color data from 6 different individuals. So the day before the solar eclipse, we just get a normal recording of these animals. They emerge hours after sunset and retreat hours before hours after sunrise and retreat hours before sunset. Responded strongly to rain though. But the next day there was this huge decrease in light intensity just because of the solar eclipse. And as you can see, these ground squirrels, they didn't respond at all. However, they did respond to a rain shower a little bit later. And the next day there wasn't any change, which is a clear, another anecdotal but clear indication that these birds are not are not directly influenced by light, but that their clock keeps them in the right time phase. Okay. Another example I'd like to show you is with bats. Bats are strictly nocturnal. They're nocturnal because of predators, and I have to also say, like for these ground squirrels, they also are so precise because they have to stay away from predators because they need visual information to see predators. And the funny thing you see there is that they all have to emerge from their roosts. And bats are social animals. They usually live with a number of individuals within one place. But they should care not to emerge too early because then they're visible for predators during daytime, diurnal predators. But the funny thing is, like, if you look at different species, all these different dots are different species of bat. Don't try to look which they are. I mean, I made one that's a Dauwentons bat, watervleermuis in Dutch, you know, the Dauwentoni. And this is the Leisler's bat, Nycteris leisleri, bosvleermuis in Dutch. But if you plot the emergence time of these bats, so here's the emergence time, here is sunset, right? And then here, this is minutes after sunset, and it gets darker and darker. And if you plot flight speed here, then you can see that fast flying bats emerge very early, and slow flying bats here on this side of the graph emerge very late. And there's this line actually bordering these bats. And you can ask, what are these bats here? Well, why aren't they on this line here? But these bats are slow flying and may emerge relatively early, but they're very agile. So in other words, these bats, fast flying bats or agile bats, are less afraid of predators. So really predation is driving the emergence time here. And then you can ask yourself, where's the clock then? But the clock actually tells them when to start checking when to fly out. And what all bats do when they emerge is they get restless. They want to leave the roost. They all gather at the entrance of at the exit of the roost, and then wait until the ambient light levels drop beside a certain threshold, and then they fly out. And it's the clock actually that drives them to express this behavior. Okay. Hang on. Clocks are ecologically important, but so with, like, is this really true? I mean, so like, do these clocks really rescue like these animals from predators? Right? Does it make them live longer? Does it increase lifespan, and also does it increase fecundity? Fecundity as in getting more offspring and getting more copies of your genes in the future generations. And that is sort of like lifespan and fecundity is usually caught in ecology in the term fitness. Can you actually test for this? And that is not so easy, but I'll show you some examples of this. So first of all, and Joke has already explained to you quite a bit about this, I have to tell you that the many organisms, but mammals and birds, they have a central circadian pacemaker. Mammals, it's most clear because it's located in one nucleus. Of course, they have peripheral pacemakers, but the central pacemaker is the boss of all pacemakers. It's called the suprachiasmatic nucleus, that links closely to the optic nerve. It, of course, takes light input, as light is the main driver for synchronizing and training circadian rhythms. Don't memorize everything on this drawing, but just keep in mind there's a central circadian pacemaker that drives circadian rhythms. And what you then can do is actually see what happens to animals if you cancel this central circadian pacemaker. And in mammals and mice or squirrels, for example, you can do this by inserting a tiny needle in the brain and very carefully locating this suprachiasmatic nucleus, and then heating the tip a little bit, and in that way you actually destroy the central circadian pacemaker. And if you then test these animals, if you did it correctly, and if you then test these animals for circadian rhythms, then you can truly see that these rhythms actually are lost. I have to say, mostly in absence of time cues. Now, already a while ago, Patricia de Corsi in the US, she did like a field experiment with chippings. Chippings are like the European ground squirrel, it's a whole different family, but are diurnal mammals. And she lesioned those animals. So she put a needle in these animals, and she destroyed the suprachiasmatic nucleus. For some individuals, she did so, and for others, she just inserted the needle but didn't heat the tip. So you had a sham-operated individual, and some she left untreated. As I explained to you here, these animals are diurnal, so they should be normally active in daytime. And here you see activity records, again with colors, when she could actually using these colors for tracking the activity. Here you see the activity for a number of days for different squirrels here. And you see this activity is really sloppy. And what you also can see is that these animals that should normally be active in daytime express a lot of activity during nighttime, which is obviously not in their ecological interest. However, control animals that only had a sham operation, only had a needle without heating the tip, without destroying the central circadian clock, do not show any activity during night. Now, so the rhythms are even in an environment where you have the natural light dark circle, the rhythms are compromised. Now, if you then look at the predation of these animals, how often they're caught by predators, then there's a clear difference. So these are the lesioned animals, these are the intact animals, and as you can see, many more lesioned animals are killed than intact animals. And then if you look at the activity, just like the graph I just showed you, then actually there's a graph that actually shows this. There is a lot more activity at night, during the night, compared to the intact animals. And if you then look at the lifespan of these animals, then you can see that the days of survival are compromised in the SGM lesioned animals. So this is the percentage of lost animals that are lost to predation. And as you can see, the number of these animals actually goes up much more quick than the sham controls those animals with only an operation without destroying the center, the pacemaker. And surely, if you compare to the untreated control animals. Okay, well, so this is where the clock of an animal actually has been compromised in a natural environment. What you also can do is not change the clock of the animal, but you can change the environment itself. And of course, that's very difficult to do outside. But what you can do is, of course, change the lighting environment in the laboratory. And that's what Ursula von St. Paul and Jürgen Aschoff did already even a longer time ago. And what they did is they took flash flies, they kept them in the laboratory, and they also looked at survival. And they kept them at a 20-hour cycle, a 24-hour cycle, and a 28-hour cycle. And obviously, these flash flies are used to live in a 24-hour cycle. And they looked at how well these flash flies actually did. And if you then look at the survival curve, the number of flies alive, starting by 100% going down here, then it's very clear that the number of flash flies in the 24-hour environment, in laboratory environment, actually live longer compared to the 20-hour and 28-hour environments, the flies in the 20 and 28-hour environments, clearly showing that that a clock that runs in pace, a resonant clock, so the 24-hour clock is resonant, the 24-hour clock in the 20 and 28-hour environment is dissonant, it doesn't resonate, So if you are in this situation, that really shortens your life. In Carl Johnson's lab in the US, a similar experiment was done in cyanobacteria. I'm just showing this example that this goes for many more species. So what this did is they raised two different strains. One strain that actually with cyanobacteria it's much easier to create mutant animals. So they raised a 20-hour strain, so a strain with bacteria that actually has an endogenous circadian rhythm of 22 hours, and another strain with an endogenous circadian rhythm of 30 hours. And they kept them, these both strains, in a laboratory environment again, with an 11-hour light and 11-hour dark cycle, which means 22 hours in total. And then if you look at the consecutive generations and you look at the number of these bacteria in the cultures they actually used, then it's very clear that in a 22-hour environment, the 22-hour rhythm strains do much better than the 30-hour strains. And conversely, if you put the 22-hour and 30-hour rhythm strains in a 30-hour environment with 15 hours light and 15 hours darkness, it goes the other way. So the green and blue line are now all the way up here. The number of bacteria in these media went highly up, whereas the strains with the 22-hour rhythm, two different strains, actually almost went extinct in these cultures. Okay. Now, how about mammals with a dissonant clock? So you could do the same experiment as done with the flies with different with mammals, but funny enough, already a while ago in Mike Menke's lab, a mutant hamster was discovered. It's called a tau mutant hamster that spontaneously had an endogenous circadian rhythm of less than 24 hours. And funny enough, so the wild type here, the normal hamster, the Syrian hamster, by the way, shows a normal 24-hour rhythm. So here there's a light-dark schedule, but here it says dark. From here onwards, everything is dark. But you can see those animals just continue with a nice 24-hour rhythm. The heterozygous, so the individuals with the heterozygous carrier of the mutation had an endogenous rhythm of 22 hours. And as you can see here in a light-dark cycle, they happily synchronize to the 24 hours, but as soon as there's no time information in a dark environment, they revert to their 22-hour rhythm. As you can see, it's shorter and shorter and shorter. And here the benefit of a double plot actually shows, right? It's 28 hours, but this rhythm actually crosses the 24-hour line here, which makes life a lot easier. And you can see very clearly that this is a very short rhythm. The homozygous mutant with a tau on both sides has even a shorter rhythm. So it has 20 hours, and this line of behavior actually is even shorter. Now, they did experiments with these hamsters. So they look at the longevity of these hamsters, or how long they would actually live. Because these hamsters are immediately very interested. I mean, it's very interesting, right? Because you know, what causes you to die, right? I mean, what is it, the number of heartbeats, or the energy you metabolize, or whatnot? So they kept them in the lab and looked at longevity. And what happened actually was that if you look at the lifespan here of the wild type, here, that's the 24-hour hamster. Here is the heterozygous mutant with the 22-hour rhythm, and here you have the tau, the homozygous mutant with the 20-hour rhythm. Then, to their surprise, actually, the lifespan of the heterozygous hamster was actually compromised. So the wild type lives relatively long. The homozygous mutant lives also relatively long, not statistically different from the wild type, but the heterozygous mutant actually lived shorter. Now, how could that actually be the case? And well, I'll show you one more slide. I made an animation for that. I'll show you in a bit. So how would that be possible? I have to say that there's later on, so Tommy Martino did some work years later on the health of these different strains. So again, the wild type, the heterozygous and heterozygous hamsters, both heterozygous. And here on this axis, actually, you see the heart weight relative to body weight, and and that's actually a measure for cardiac problems. So the heavier the heart gets, the more it needs to work. And as you can see in the heterozygous, the heart weight gets relatively much greater compared to the wild type. But funny enough, if you lesion the central clock, the suprachiasmatic players, again, like remember the picture I showed before, then you actually rescue the cardiac problems that these animals actually get. So it has to do something with rhythm. Now, I made a small animation, and I hope you will understand it, specifically for this talk. I like it a lot myself. I hope you like it too. So here's the three different genotypes, and I just like try to depict there. Can you see this actually? Yeah? Okay, very good. So it doesn't end well for the middle one, that is the heterozygous hamster there. There's a happy end for the wild type, and there's a happy end for the homozygous mutant with the 20R. But have a look at the actograms. So the wild type, of course, has no problem because it can synchronize to the 24-hour. The full mutant, the homozygous mutant, actually does what it likes. I mean, the endogenous circadian cycle actually prevails. It just reverts to its own rhythm. It's more active in the dark, that is to say. I mean, it's a nocturnal species, but still it runs on its own rhythm. But here the heterozygous actually does entrain. So like the 22-hour rhythm is not all too much off the 24-hour rhythms, only 2 hours, and here it's actually 4 hours. So it's still synchronized. And in fact, this animal has a permanent jet lag of 2 hours. So actually that is most likely the cause of its problems there. Okay. Now I'm gonna speed up a little bit. I'm just gonna tell you a little bit about the activity rhythms of the same tau mutant animals in the field. So you can actually, this was all in the lab. So how would it now work in the field? So how would a dissonant clock in a more natural situation affect survival and fecundity? And for this we used mice, because at the time, years later, these mice with the same mutation were actually produced the same mutation compared to those hamsters. Again, you have here the wild type, which has an almost 24-hour rhythm. Here you have the heterozygous with a 22-hour rhythm, and you have the homozygous with a 20-hour rhythm. And what we did is we released them in enclosures in a Mendelian ratio. So we have in each enclosure 10 homozygous, 10 wild types, 20 heterozygous and 10 homozygous, which is a Mendelian ratio. So the mutant allele is about 50%, the mutant gene. We kept track of these mice just by injecting them a microtransponder and logging their activity around in this enclosure when they actually went to the field. So we could actually track the rhythms. And regularly we caught these animals, but I'll show you first a small footage of predation. So these mice were exposed to natural predation. This is an owl that sits on one of the feeders, and there's a red-tailed hawk that actually catches a mouse. Here you see it flying again. Here it catches a mouse here. So there was quite strong predation. So there's and we registered them continuously. And we kept track of the genes in that population, and we also kept track of their rhythms. And of course, when you catch a mouse, you take a little bit of a small DNA sample, and you can actually tell what genotype it actually has. So first of all, we track the rhythms here, and here you can see the rhythms of these different mutants. So here's the wild type, mostly constrained to the dark phase. This is a long record. You can see the summer here, the early summer here. This is the 22-hour mutant, and it's the 20-hour mutant. And in contrast to what happened in the lab, if we looked at lifespan, we saw a compromise for the tau homozygous tau mutant and not for the heterozygous. And what we also saw was a reduction, a gradual reduction in the mutant allele, telling us that a dissonant clock can be problematic for you if you're in a more natural environment, if you have to survive and if you have to reproduce. Okay. Now, lastly, I'm gonna give you an example about the importance of yearly timing, and that is an example from close by here at NEO, where we look at the year cycle of great tits. It's a passerine bird that lives in the forest here and is highly dependent on a food peak in spring. And the food peak mainly consists out of caterpillars or winter moth, but the interesting thing here is that the bird needs to anticipate this peak in food. And it needs to anticipate because it's a bird. It needs to produce an egg, it needs to lay the egg, it needs to breed on the eggs, and it can only use the food peak at the moment the young actually can be fed food. Now, it's very difficult to experiment with year cycles, of course. That's really, really difficult, especially outside. But there's sort of a live experiment going, and it's climate warming. And the interesting thing with climate warming is that the insects, the food peak biomass of caterpillars, respond very, very directly to temperature. Whereas these birds, they cannot. I mean, they have to sense day length, they have to sense and also temperature, but they have to anticipate. And as a result, these birds, so these birds actually have only succeeded to advance a little bit, only about 1 week over the last decades, whereas this food cycle shift has advanced over 2 weeks on average. And that causes a problem for these birds. So these birds that are actually too late, they don't have access to the food peak anymore because they have their chicken too late, and these chicken have truly a lower chance on survival. Okay, then one last example, and that is collective breeding. Actually, I'm gonna skip this example because I'm running out of time. But I'm sure that I convince you that clocks are highly important in ecology. And I would like to end my talk with some information on the embedding of the clocks and ecology in the BioClock project, because it may be strongly compromised in modern society, and it all has to do with our human habit to illuminate our nocturnal environment. For a billion years, the Earth has been spinning around and was light in daytime, dark at night, sometimes moonlight, of course, and starlight, but it was never as light as when the Anthropocene started, with us planting light posts everywhere and illuminating our environment. And the most strongly driving factor in there is our economic growth. And I also like to show you this picture very briefly, but this is it's actually a harsh view of the consequences of economic growth, or the opposite. So this is North Korea, which is completely dark, and this is South Korea, which is completely illuminated. So anyway, you may envision that illuminating the dark sphere may disturb the natural light-dark cycle and thereby compromise the circadian clock. Well, I'm going to skip this a little while. I'll briefly tell you a little bit about this, but artificial light has a very strong direct impact on species. Here you see birds attracted to light, and actually if you leave these lights on, these birds actually exhaust themselves and may even die. And this is the same for insects. There's also a direct consequence, for example, for habitat loss. And that goes for bats, for example. As I told you, these bats are very nocturnal, and especially these slow-flying bats that emerge very late from their roosts. If you illuminate a patch of forest, they just lose habitat. But these lights also may have an impact on activity rhythms. So here I show you the activity rhythms, the foraging rhythms of different of a bat species, or pipistrelle bat. And as you can see, we have a big experiment here where we experimentally illuminate patches of forest with different colors of light, and we measure the bat activity around these different lights continuously. And I've plotted it here for I made for different the nightly profile for these different light colors. And as you can see, there's foraging in the dark, but mainly a peak in the evening. But as soon as you apply light there, then bats can first of all forage a lot more there because of the attraction of insects, but also at different times of night. And that's actually also dependent on the light spectrum there. Another example of the impact of light at night is for birds that sing in the morning. So here, just like the emerging bats, you see the time before sunset. Here is sunset, and this is the night. So the deeper the dots are here, like this robin, the earlier they actually sing, and this is the date in April, so the 1st of April to the 18th, and that repeats itself for these different species. Now, the dark dots are a natural dark environment, but the light blue dots are an illuminated environment, which clearly shows that these birds actually start singing much earlier if there is light in their environment. And it also shows that if these birds are relatively early in the morning, their advancement is much greater, presumably because they're actually more sensitive to light. Now, a big question there is whether these, whether they actually it's just an effect of an activity pattern, or whether it really affects the clock, whether actually these birds living in these different environments actually have a different clock. And one of the first signs of this is was done in an experiment by Davide Dominoni, who took blackbirds from urban and rural environments, from forests and cities, into the lab. So he first showed that city birds are actually active much earlier than forest birds in the morning, relative to the civil twilight. Again, you have here this is early in the morning, this is later in the morning. But then if you measure the endogenous circadian period, so their own rhythm, if you put them in the laboratory, then you see that the city birds actually have a shorter period length compared to the forest birds. And that's a true sign of a change to their circadian clock. Now, for the sense of time, I'm just gonna skip those slides. If you want, we can I can show you them in the at the end of my after when we chat after my talk. I just show you a few examples of seasonal rhythms that are affected by artificial light at night. This is a photograph a friend of mine sent me, actually a colleague, Christian Bott. These are light posts here, and these are birch trees here. And actually, I don't think I need to explain what actually happened here, but it clearly shows it has an impact on the leaf coloring of those trees. And then I think one of my last slides here. Hang on, just need to check here for a bit. Yes, exactly. Is one slide on the impact of seasonal rhythms for birds breeding at our experimentally illuminated sites. What we do is we kept track of their laying date. We put up nest boxes there for crate dits, You can easily open them, you can look at when those birds actually lay their eggs, and then you get the lay date. So the higher up here in the graph, the later these birds actually lay their eggs. And as you can see in the green and in the white illuminated spots where these light posts actually are, these birds lay their eggs early, and that's a clear indication that lights have an influence on the yearly timing of these birds. Okay. Well, there's also food web effects there. I'm just informing you, there's relatively little known. So Jonathan Benny has done microcosm experiments. There's some more experiments with bats and insects, but this is still a highly underexplored field here, and especially how the clock actually interacts with these different food webs. And that is actually something that we're trying to tackle in the big BioClock project, which is actually so exciting about this whole project. And it's very important because changes in the daily rhythm, changes in the nightly rhythm, depending on how bright our skies are, how brightly illuminated we illuminate our skies, the more likely these rhythms actually are disturbed, and the more likely species get into trouble, and more likely there's an impact on biodiversity. So clocks in ecology, we really should care about them. And with this, I'd like to end my talk, and I'm happy to take any questions.
Thank you very much, Kamiel, for this very exciting talk. Very nice. I was just keeping track a bit of the questions, and I think we have one more or less comment from Connie Berge. I'm not sure if it's just a comment or a question. That's why I would ask you to unmute and maybe specify what you meant in the comment. Connie, would you like to do that? Yes. I think you're not muted now. Yeah.
Okay, well, I can't see you yet, but
we can hear you. We can hear you, Connie.
Okay. My remark is the statements you are using about seasonal. Is that referring to the Netherlands, to West Europe, or I think this useful to a disclaimer about which location you are using. And the other remark is about the mechanical clock time. If you are saying 0 and 12 hours, for instance, today, when it is 12 o'clock, it's really 10 o'clock because of the time zone we are using and the extra clock change. So I think this useful to or to disclaim about which time you the time you are referring to, or using you can also show the negative consequences from using the unjust mechanical clocks today especially in Western Europe.
Okay, thank you, Connie. To explain a little bit, so actually in most of my, if all of my examples, I wasn't really referring to the absolute clock time. So it's therefore, I think in all cases, it's not all too relevant what the absolute clock time is relative to the activity, the expression of activity. It's more like to see the relative differences between the different expressions of activity, right? So you could as well plot your activity relative to astronomical time, but then the shifts in activity would actually look the same. So that is not so crucial here.
Okay, thank you.
And then we have another question from Irtiza Tahir. Can you suggest that these clock rhythms can predict life expectancy? That's the question for you, Kamiel. And if it's not specific enough, we can also ask Irtiza, sorry for misspelling, to unmute herself or himself and ask the question.
Yeah. So that's a nice question indeed. So like it truly looks like if there's a too much of a discrepancy between the your internal endogenous circadian clock and the external clock that is actually imposed on you, so like the clock you have to live with, then that in that case actually compromises your lifespan. And for hamsters, this has been researched by looking at many different health problems in these hamsters. I showed an example with heart problems, but they also check kidneys and blood pressure and many other parameters. In other words, like if you happen to have a rhythm that actually is much different from 24 hours, it's probably in some cases better to not to violate it and to live your own rhythm in that respect.
Hope this answered the question. That was it. We have a lot of thank yous and congratulations for your talk in the chat. So we still have 2 minutes left. In case anybody has a question, you can raise your hands or unmute yourself and ask Kamiel. We still have the time. Kamiel, maybe you can stop share screen.
Yeah,
and I
sure.
see better.
Let me see.
I think I can also stop it.
Yeah, I have to get back to my.
I will just.
I have to find my my Teams environment back, but it's seems to have disappeared.
It's fine.
Oh yeah, right. It's on my other screen. Okay, now I'll stop sharing.
Now we see each other all. Yeah. So I don't know. It seems there's not any other questions. I wanted first, before everybody leaves, to thank you all for attending. And if in case you are interested to know more about it, just check the BioClock Academy website and the BioClock website itself. We will put all recordings there. So if you missed the very first session, you find it there. Yes, and we will continue with this with only the BioClock trainees, so you can stay in this Zoom link, and all the other people are then we invite you to leave the chat politely. Thank you very much for attending. And